Hello everyone, let's start with the series roadmap to become competitive programmer 5 star. So in this video I'll be solving 44th problem, complementary strands in a DNA. So I think bio students knows what is complementary strand. So don't worry, it is very easy only. I'll try to explain you. You're given the sequence of nucleotides of one strand of DNA through a string S of length n. S contains the character A, T, C and G only. Yeah, as we know, we have only four sequence of nucleotides. A, T, G and C are basically adenine, thymine, cytosine, guanine. So yeah, these four nucleotides we have, and check if chef knows that A is complement. We know A is complementary to T is complement to A, C is complement to G, G is complement to C. So using the string S, determine the sequence of the complementary string of the DNA. So basically, okay, first line of integer contains T, denoting the number of test cases. So this is T. C in T while T minus 1. This will take care of all the test cases. I'll call one function for every test case. Let's call it as, name it as solve, whose return type is void and does not have any parameter. First line of input contains integer and denoting the length of the string. So first line contains n, which is basically size of the string. We'll take one variable string, we'll call it as DNA, and we'll input into DNA the string, and its length is n, that they have said. And they've given four test cases. First test case n is 4 and ATCG. So let me try to explain you. Sorry, I'll have to move this here or else it will be not good. What was it? ATCG. So and the given is 4, which is length of the string. If you count 1 2 3 4, 4 is length of the string. So as you know, DNA is kind of in a human body we have like this. So here we have nucleotide, a chain of nucleotides. So let's say A, T, G, C, something like that, in any combination. So A's complement is T, T's complement is A. We know these things, right? Adenine's complementary is cytosine, thymine's complementary is adenine. C's complementary is G, G's complementary is C. So like in one strand, this strand, let's say S, which is, let's say, this one, and this is S dash. So A will always be bonded with the T, and T will always be bonded with A. This is the condition that they're saying. Okay, it's easy only. So they have given string S, which is ATCG. We'll have to find S dash, and we know A's complement always be T. So whenever there is A, we'll have to put T. Whenever there is T, you'll have to put A. So if you think of as a DNA, A is bonded with T in the S dash strand. This is one original, sequence of ATGC, and this will be a complementary of that, and both will be coiled, super coiled basically. So that is again a bio part, but in our problem it's very easy only. Whenever there is A, you will have to put T. Whenever there is T, you will have to put A. Whenever there is C, you will have to put G. Whenever there is G, you will have to put C. So this is our answer basically. TAGC. TAGC, yeah, this is our answer. In the next test case again they have given GTCC. Length is 4, but sequence is GTCC. Guanine, thymine, cytosine. So S dash will have to find. When there is G, you will have to put C. When there is T, you will have to put A. When there is C, again you will have to look into this and put it. When there is C, you will have to put G. So CAGG is our answer. So yeah, CAGG is our answer. So let's take other example. We all have AAA and length is 5. So n is means length of the string is 5 and all has 5 A's. So S dash is basically the complementary of that. Whenever there is A, you'll have to put T. So this is the thing. So yeah, that is the thing. T you'll have to put. And when there is TSC, let's consider this also. You will be very clear with that. What was it? Tag. It was TSC. So n is 3, basically length of the string. S is T A C. S dash is when there is T, you'll have to put A. When there is A, you'll have to put T. When there is C, you'll have to put G. It is ATG. Yeah, it is ATG. I think you guys got to know how to solve it. So it's easy only. First, what we'll do is we'll take string. Let's name it as S only. Let's don't name it as DNA. And we'll have to find string S dash. So for dash, we'll put something like S underscore. We'll put, okay. Or we can say complementary of string S. CS will take. That is right now empty string. We'll take one loop. We'll be iterating through each element of the string S. If we find S of I to be A, and all they have given capital letters or what, let me check. Somewhere they have said or no, but in the example we are seeing it is capital letters, so I believe it will be in capital letters only. So whenever there is A, you will have to insert, you will have to take complementary of A, which is T. Else if S of I is equal to T, in that case you will have to put its complement A. Else if S of I is, let's say it is G gone in, so cytosine will have to put C. Else definitely if there is like we have only 4 nucleotides A, T, C and G, so ATG is considered. So if all these conditions not met, then definitely that will be C. So for C's complement it is G, and at the end you will have to print this complementary string. So if you see TAGC, TAGC, CA, yeah, everything looks fine. Let's try to submit the code. Yeah, it got accepted. So I think that's it from this problem statement. So it was easy only. You will have to only basically focus on this stuff. Whenever there is A, you'll have to put T. Whenever there is T, you'll have to put A. Whenever there is C, you'll have to put G. And whenever there is G, you'll have to put C. So yeah, you'll have to basically build the answer string from the given string. And yeah, that's it. So thank you guys for watching the video.